What I wanted to ask you is, have you studied the structure of these molecules in terms of, the structural biology aspect?
So another great question that I love to answer because that's a big part of our research. So a big part of the chemistry part of our research is really characterizing these molecular dynamics of how the aptamer captures a small molecule. And this really started as a question during my PhD work because we had a sensor for dopamine, we had a sensor for serotonin, we had this field effect transistor. One increased in terms of the current with increasing amounts of the molecule, and one decreased. And that was really strange considering the fact that the two molecules are very similar in structure, similar molecular mass, same positive +1 charge at the pH we're measuring in. And so it's like, okay, it has to be the DNA doing something in terms of such opposite dynamics. So we use a lot of spectroscopy, for example, circular dichroism spectroscopy, that allows you to monitor how the DNA folds in terms of the secondary structure. But these are solution-based techniques where everything is just floating around in solution. So it doesn't really mimic what happens when you tether it to a surface, where now it has less degrees of freedom. Now it's tethered. And so we have other methods like quartz crystal microbalance, where you can assemble molecules on a surface. And this method works in that it's a piezoelectric crystal, meaning that if you apply a voltage, it oscillates at a certain resonance frequency. You can assemble molecules, and then with higher mass, you have less frequent frequency changes. And then what's really cool is that you can also see compression versus elongation of the DNA backbone, because DNA is very hydrophilic, so it has a lot of water molecules attached to its backbone. So when it's compressed, you kind of get rid of water molecules from the surface, whereas when it's elongated, you have more water molecules attaching. So there's different ways to characterize it in these kind of indirect ways, such as height changes. But we also have a collaborator at IIT, Dr. Dennis Garoli, and his postdoc, Ali Douaki, has been able to do molecular dynamics simulations to finally visualize the structure switching. And I think that was really a moment that was super exciting, where we can finally see, oh, this is how the dopamine aptamer folds. This is how the serotonin aptamer elongates. This is a possible binding pocket of where the molecule gets caught inside the sequence. But of course, with these simulations, you get a variety of structures that are the most thermodynamically stable, likely structures. So we also have to do experiments to validate them. So a potential for that is FRET, Forster resonance energy transfer. You can put two dyes in different locations of the DNA backbone. If they're within a nanometer from each other, you have quenching. If they're further away, you see fluorescence. So that way you can also see kind of what part of the backbone is coming close or not. And these are experiments that you can correlate with what you see in the simulations to say, this is the most likely structure. So now I would say, have about 6 to 10 different ways in which we can characterize the aptamers. And it took a very long time even to know how and what to do. But I think it's a really exciting start, because now we have the tools. We can say, okay, we have a new sequence, but we know how to kind of go through the protocol to characterize it, to predict how it will behave inside the sensor. And perhaps there are some structural switching dynamics that are more optimal for certain sensors. These are the things that we want to figure out with our new kind of way of characterizing the aptamer-target interactions.